So welcome everybody, bienvenidos, bienvenidos. Welcome Professor Roy Osseri, Vice President for Resource Development, who will officially open the event in a few minutes. Thank you for joining us. Also with us is our main speaker for today, Dr. Meital Oren, from the Department of Neurobiology. We look forward to your talk. I would also like to thank Mario Fleck, Dr. Mario Fleck, who will manage the Q&A session at the end of the talk and will conclude the series with closing remarks. You are welcome to send questions via the Zoom chat. And now I would like to hand the microphone over to Professor Roy Osseri. Please, Roy.
Thank you very much, Danny, and thank you all for joining in, all the way from far away Latin America. I think that, on the one hand, I'm very sorry that we can't have you here on campus, and I can't travel to Latin America and meet each and every one of you personally. But on the other hand, I think this situation has taught us how to communicate without travel. And that means we can engage in seminars like this one, and I think that's a great opportunity. Danny asked me to give a short update on things that are going on at the Weizmann Institute. I think that the biggest news of the last few days was the signing of a memorandum of understanding that we signed, actually also using Zoom, using a digital platform, with the United Arab Emirates, actually with a university, with the Mohammed bin Zayed University for Artificial Intelligence at the United Arab Emirates. That was 3 days ago via online means. This is, I think, the first collaboration agreement between an academic institution in Israel and in the United Arab Emirates, and perhaps even in the entire Arab world, although I would be careful. I'm not sure if that's true. We're very excited about strengthening the ties, the scientific ties. The Mohammed bin Zayed University is a very unique university in a sense that it's a research institute solely dedicated to research in artificial intelligence. They were able to recruit a very broad and talented group of scientists that came to the Emirates in order to perform research there, and they have one of the biggest computers in the world, certainly much larger than any computer cluster we have in Israel. I think that's a major breakthrough, and especially since, as I'll tell you in a couple of minutes, AI is not really about computer science, but AI has a significant impact on our ability to do research in any domain of science. And this signing of MOU has caused a lot of excitement, of course, at the Weizmann Institute, but also around Israel and around the Arab world. And I'm going to show you just two very quick examples of media coverage. So you can see here a story in Haaretz, one of the leading newspapers and online portals in Israel. And this was covered by all the Israeli media, as well as a cover story in Asharq al-Awast, which is a leading newspaper in the Arab world as well. So I think it's causing excitement everywhere. I was interviewed to the BBC radio this morning, and people are certainly excited about the possibility of performing good for people in Israel, in the region, and humanity, as we often say. So, except for this particular agreement, I also wanted to update you on 3 flagship projects that we are pushing forward very hard. One of these flagship projects is, again, AI, so it's very intimately related to our agreement with Mohammed bin Zayed University. We understand, the world of science has undergone a profound, I shouldn't call it revolution. I think it was an evolution, but it's an evolution that transformed the way we do science. And that evolution has to do with the huge overload of data that our scientists have to deal with. And I can maybe connect to your own personal experience if I'll ask you to remind yourself how your life looked like 25 years ago or 30 years ago when you had no internet, no smartphones. When people had to call you, they would dial on a phone, and you maybe had to deal, you connected with people maybe 4 or 5, maybe 10 times a day. Today, your smartphone is an incredible information channel. You're overloaded with information through WhatsApp, text messaging, phone calls, emails, social media portals. It's very difficult to deal with this overloaded information. A similar evolution happened in the scientific domain. Scientists today, and it doesn't matter if it's life sciences, genetics, neurophysics, neuroscience and brain, astrophysics, or material science and chemistry, any domain in science, the amount of data that we harvest is enormous. But in order to turn this data into knowledge, to turn this data into scientific findings, it's no longer sufficient to use a pencil and a pen. We now need very strong computing resources to deal with this data. And today, the most efficient and the strongest tools that we have come from artificial intelligence, come from the world of machine learning. And that's why we understand that in order for ourselves to advance forward as a scientific institution, we have to make these tools available to our scientists. And we'll do that by doing two things. One of them is making sure that our scientists have the access to the best and the most cutting edge high performance computing hardware there is, either by building a facility on premise or by allowing our scientists access to cloud services. The second pillar on which this initiative would stand is integrating AI specialists, people who understand computer science, into our departments and faculties, people who would help physicists like myself or biologists and neuroscientists like Meital, Oren, Suiza, to take advantage of these algorithms and push the research forward. This is a very large, ambitious plan. It would cost about $100 million, and we're working on this full time. The second flagship project is a flagship project that we call the Frontiers of the Universe. The Frontiers Institute would be an institute that would advance research in fundamental physics, from particle physics, asking questions like, what is dark matter composed of? You know that the universe, 70% of the mass in the universe is made out of matter that we're unfamiliar with. It's made out of particles that we don't know what they are. So particle physicists are trying to find these new particles and identify what 70% of the universe is made of. Astrophysicists are also trying to look at the structure of the universe, how stars are born, how stars die, the way these big cosmic explosions with which stars die send ripples of gravitational waves into the universe. And in fact, within this Frontiers Institute, we are going for the first time to have a Weizmann Institute space program. I think it's the first time that any academic institution in Israel is managing its own space initiative and space program. The first sparrow of this space program is going to be a satellite that will launch to space in 2024. It's called Ultrasat, and it will identify cosmic explosions and look for signatures for gravitational waves. You see that these two ends of studying the smallest things in the universe and the largest things in the universe actually connect. And just to give you a bit more inspiration on it, I'm going to show you the new physics building, which would be home for this Frontiers Institute. It's this building that you see here. And for those of you who are familiar with the Weizmann Institute, this is where the particle accelerator is. and connected to it is the old physics building. This would be the new Frontiers Institute. And in it, you can see this would be the satellite control room. As I said, Ultrasat is only the first satellite. We'll have a series of satellites that would be sent to space with scientific equipment on it. The control room would be at the Weizmann Institute, and our plan is to build an auditorium to which we can bring school groups and classrooms that would visit the Weizmann Institute. They would get educational lectures on astrophysics and space. And after the lecture, we can open the curtain, we can open the screen, and we'll able to see the satellite control room in action. And we really hope that this would be, would have an Apollo effect on a new generation of kids, either from Israel or visitors from around the world, and would inspire them to become scientists. The last, but certainly not least, flagship project that we're pushing forward has to do with the subject matter of what you're going to hear about today. And this is our Brain and Neuro Research Institute. This Brain and Neuro Research Institute would consolidate and advance brain research and neuroscience, neurobiology research on campus. The reason we chose to have our next flagship in the neurobiology research is because if you examine life sciences for the past 20, 30 years, you'll notice that life science was transformed by the genetics revolution. And anything that has to do with genetics was really completely transformed. And one expression of this transformation is the way cancer, for example, is being treated today. If you look at the way cancer was treated in the 70s or 80s, because the understanding of the genetic mechanisms behind cancer was still lacking, the only way you could try to medicate cancer was by poisoning not just the cancer, the malignant tumors in your body, but in fact your entire body. And people still treat cancer this way with chemotherapy and radiation. But today, because we understand the genetic mechanisms of cancer, we can actually attack cancer tumors by targeting specific genetic sequences with biological medications. One area of biophysics did not have that Watson and Crick moment yet and was not transformed fully, and that is neurobiology. I think that if you look at the place in life sciences where the big revolution is still ahead of us, that's certainly neurobiology. The Weizmann Institute is positioned at a very good starting point to make that scientific revolution. We have an amazing group of neuroscientists. And I think one of the most important things to remember is that neurobiology is a highly interdisciplinary field of research. If you want to make impact in neurobiology, you have to combine the efforts of biologists, of chemists, of physicists, of psychologists and psychiatrists, of computer scientists. In fact, the field of brain research is intimately related to the field of artificial intelligence. In both cases, the models behind the best models we use today rely on networks of neurons. And that's why we believe that if we'll build a home, again, that would be a building, a house for all the neurobiologists and all the interdisciplinary research of brain and neuroscience at the Weizmann Institute, we will be at a very good, at a promising position to drive, to push forward that next revolution, next scientific revolution. And when we'll do that, maybe similarly to cancer, where today medications are more efficient, more precise, and can target different specific tumors in cancer effectively. Also in the case of, for example, neurodegenerative diseases and diseases that have to do with aging, again, we will be able to treat these better, more effectively, and with higher success rates. This is, again, a big initiative. It would cost about $200 million, $100 million for the building, and another $100 million for operation. And I know that our Brazilian association has generously stepped forward in order to support us on this mission. So thank you very much for that. So with that, I'd like to end. I think ending with a brain and neuroscience flagship is very befitting, since the lecture you're going to hear now is exactly on that.
Thank you very much, Roy. Thank you very much. It's a great opportunity to hear you today. I just want to say, before we continue with Dr. Meital Oren Suiza, to welcome Raquel Sibob, that she came back from the maternity leave, and all of us, we are very happy. Raquel, maybe you don't recognize her because she cut her hair, but it's the same person. Now, Dr. Meital Oren Suiza will introduce, you will introduce yourself, I understand, Meital, please.
So let me just share my screen. One more. That's it. Everybody can hear me, right? It's fine, Danny?
Yes, perfect.
Perfect. And you told me to time myself because scientists like to talk and talk and talk. And you know what? You're right. So I'm going to start the timer. Thank you very much, Roy. Thank you, Danny, and all of you. It's an honor to spend the next 15 minutes of my talk and then hearing what you have to say, and we can, I'm looking forward to the questions as well. So it's good morning and afternoon, right, in different regions. And so I will start by introducing myself, and I think I have a saying that we have in Israel, which I'm sure you all know, that the Jewish mom tells her kids, you can be anything you like. You can be a doctor or a lawyer. And I think my mom eventually is, I don't know if that's what she meant, but I think she is proud. And as a kid, I grew up in the periphery of Haifa, and I loved sciences. I really like chemistry. And so I got to the Technion because of the chemistry, and I fell in love with biology in the end. And so I did my undergrad and my PhD at the Technion. And I was working on aspects not related to neurobiology, but fertilization, and specifically the fusion between the sperm and the egg. I think it's something that is common to all of us. This is how we're conceived. But I became fascinated with neurobiology during my PhD, and then I went to do my postdoctoral work, starting to investigate the variation between the sexes in Columbia University in New York, and that was a fabulous time to be in New York. I don't know if we can say the same for the last couple of months, but I was very, very happy and lucky to come back to Weizmann. I live in Weizmann as well, and I work here. And I don't think it's a cliche to say that this is really, you know, a dream coming true and exceeding my expectations. And I'm very, very happy to be here. It's a wonderful place to work in and also to live in. So I think this is a nutshell in a nutshell about myself. Okay, and so the question we're interested in the lab, and I'll give you a taste of the introduction and, you know, the relevance to diseases and what we do, is how does the sexual identity, or more specifically, the genetic sexual identity, whether our chromosomes are male or female, how does that affect the connectivity or formation of patterns in our brains? And when we think about the sexual identity, it's just one layer of an identity that the cell gets, right? Because many, many factors can affect our cells, but we're interested specifically in how, you know, the genetics of our sex affects individual cells in the brain. So if we think not about humans, of course, we are important, but if we look around us in nature, sex differences between animals in the animal kingdom exists just about everywhere. I think there are many, many examples across fish, mammals, birds, whatever you want, spiders as well. But we mostly think about the differences in the morphology, in how they look. But we know that that's not the only place where we see differences. There are differences in morphology and physiology, but also in behavior. And if we think about it, behavior, of course, stems or starts from the nervous system. And this is what we're particularly interested in the lab. And I'll just show you a nice and funny movie to illustrate the differences in behavior. We're also seeing morphological differences, but I want to focus on the behavior. So look at these two birds. It's the two sexes of the same species. The fancy one is actually the male, and it also sings, which we will not hear, but look how nicely it looks and how it dances in a very orchestrated and interesting way to attract the female. And I want to highlight an important point here. We all know about Darwin's first law of natural selection, but we don't always think about his second important insight, which is sexual selection. And that's how animals are shaped in order to mate and then transmit their genetic material into the into the next generations. And if you look at this male, it's very clear that sexual selection is much, much stronger than natural selection, because this is a very easy prey, right? It's very fancy, it's clumsy. And so I think it shows how strong is sexual selection, even much more than the natural selection. And it's important to remember these insights when we think about how sex differences are brought about. And we should know that sexual selection it's not the only driving force, but it's the major one for generating sex differences. So I think that should always be in the back of our minds. There is a lot that we know, there's a lot that we don't know. And I'll just say that if we think about, I told you that we're interested in the chromosomes, and that's the genetic component that we're talking about. And we know that the way the biological sex affects our brain is in two different ways. So I'm talking about the sex chromosomes, and males and females have different sex chromosomes, XX for females and X and Y for the male. And if we think how or what we know about the way that our genetic composition affects our brains, it's in two different ways. So one that we know quite a lot about is the way that our sex affects secretion of hormones. So we all know about hormones. Hormones are secreted by the gonads, and the fact that they're secreted by the gonads to the brain says that this is something that doesn't happen inside every cell in the brain, or what we call in a scientific term, non-cell autonomous. So this is done, right? It's an effect of hormones penetrating the brain and then having an effect on the brain. And we know quite a lot on that type of an effect. But what happens to the cells themselves? How is the fact that a cell in our brain, and more specifically a neuron, has an XX or an XY matters to that cell? So that is very understudied. We don't know so much about it. And this is what we're actually focusing on in my lab. Okay. What's the motivation? Why are we even interested, and where are we going with it? So I think when we think about all the diseases that affect the brain, and unfortunately there are many of those, and some of them are depicted here. So let's not talk about these two, anorexia and bulimia, because these clearly have a social component, so I don't want to talk about them. All the other diseases, and if you'll see, it's all the major diseases that affect the brain. They have a different manifest between males and females. So either the two sexes are affected in different numbers. For example, Alzheimer's, which doesn't appear here, disproportionately affects females. Parkinson's disease, male biased, 1 to 3 will be males. So I think this is one way that we're seeing differences, but not only, also the symptoms of the disease, the severity, they are very, very different between the two sexes. And when we're talking about personalized medicine, we should also consider gender-based medicine, but we're not there yet, let me tell you. We're very, very far away from even understanding these diseases, let alone understand why they're different between the sexes. And the field of understanding that gender now matters in medicine has just emerged in the last, I don't know, maybe 5 years. I think none of us have talked about it before, but now it's becoming very important. So I think the motivation for these studies are very, very clear. And we'll talk, and I'll touch a little bit in the end on Parkinson, because we are working towards understanding the differences in this disease. Okay, so just one slide to give us a little bit of the terminology when we talk about the brain. I'm sure some of you or all of you have heard these terms, but just a little bit. The brain is what it is because it's composed of these functional units, which are called neurons. So the brain is composed of many types of cells, but the important ones are neurons, and they form these networks. How do the neurons communicate with one another? they communicate via these structures that are called synapses, discovered by Ramon y Cajal many, many years ago. Synapses are either chemical or electrical signaling that the neurons transmit, and that's how information flows, and that's how circuits are formed. Neurons talk to one another. A lot of neurons will form a circuit. A certain circuit will have an input of information, and that's our sensory, what we sense, and then the output would be our behavior. Okay. But how can we look at these synaptic connections, and how can we even compare them between the two sexes if we have so many neurons? How will I know to follow a specific synapse, and how do I know that it's specific or it appears in both sexes? So for that, we utilize the tool that really revolutionized, I wouldn't say just neurobiology, but biology in general, and that's the use of a protein which glows. It's called the green fluorescent protein. Scientists discovered it from jellyfish 20 years ago, and the Nobel Prize was awarded for the discovery of the green fluorescent protein in 2008. And here you can see, so what do we do with that protein? If you take this GFP, and it's a molecule that has a nice property, if we shine it with blue light, it emits back green light. And because it's light, we can measure it with light microscopes. And if we take that protein and we tag it or combine it to any gene product, any protein. The proteins are the products of genes, and proteins are the building blocks of our cells. So if we attach it to any protein we want, boom, you can see a glowing fly, a glowing worm, or a glowing fish, or even more specifically, if we think about the resolution, if we zoom in, a glowing neuron. And now think that I can label any protein that I want, and I can follow the dynamics, and I can measure. And what's the resolution? What can we do with this tool? This is the resolution, around 200 nanometers, and nanometer is 10 to the order of 9. So that's really quite small and quite deep within a cell. So we took that GFP protein and we've split it into two. We took the molecule and we just cut it into two halves. We've attached one half to the presynaptic cell. That's the cell sending the information to a specific one. And the second half to the postsynaptic cell, the cell receiving the information. So now synapses, they have to be, neurons can form a synapse only when they're really, really close. And you can see in this nice illustration that was made for me, that only when these two halves meet, we get light. So if we see green glowing, we know that we have synapses, and if we don't, then we don't have any synapses. And this is another illustration. I have to tell you it's not accurate because information flows only in one way, but it's nice to make the point that I want to make. Okay, so how does it look? How does it look in a living specimen? You see the processes, the cytoplasm of the neurons in red, and you see the 3 dots. These are 3 synapses. Each one is about micron, so that's a 1/1,000,000th of a meter, so it's very, very small. And this is a live specimen, so we can really follow now the development in a super specific manner.
It has like, for example,
okay. Sorry, I'm hearing someone in the background. Can I mute? Because I can't hear myself.
Can you help us, please, and to mute the microphone? I try, but someone open.
I don't know how to do it.
Now it's okay.
Okay.
Now it's
yeah.
okay.
Okay, so how does it look in the animal? So we labeled the connection between these two cells. It doesn't matter, the names are for me. This is the area where I expect to see the synapses in the female, and the exact same area in the male. And you can see that this female doesn't have these connections, the green puncta, and the male does. Okay, so we know that there is a difference here in the connection between the two same cells. So it's exactly the same neurons. The only difference is that one of them is a male organism and one is in the female. Okay, and if I extend the analysis of these connections throughout development, this is in the adult. What happens in the beginning? What happens in the juvenile stages? So you can see that a really nice pattern emerges. We start from the same number of connections between these two cells, and somewhere around sexual maturation, these connections start to change. The male still forms the connection, and you can see the number of these synapses increasing, but females start to lose the connection. It's again the connection between same two cells, and the only difference is the sex. And the females now eliminate the same connection, and that's how we get a really different endpoint in development where males have these synapses and females don't. And we've done this for many, many, many pairs of neurons, and we found an interesting, a novel phenomenon, which we were the first to describe, where we get the mosaic pattern in adulthood by eliminating synapses or connections in the brain in a sex-specific manner by starting from the same template, right? We have the same cells in our brains. It's not that males and females have different cells. Yet somehow through the mechanism of eliminating these synapses, we end up with a mature nervous system that is different between the sexes. And I will not talk about it, but we also understand why when we analyze the behaviors that we're seeing in these young animals compared to the older ones. Okay, this looks, so what we wanted to know and what we've been working on for the last couple of years in the lab is how do you get this specific removal just of a certain synapse just in one of the sexes? And this looks very complicated. We're not gonna go into it, but I just wanna tell you a few highlight points from the slide that first of all, we really understood all of the mechanism, how everything happens. And I'll tell you the key highlight findings. So we found that the machinery that removes these synapses, something has to actively remove them, is a very known machinery. It's called the protein degradation machinery of the cell. And actually a Nobel Prize was awarded for the discovery of this mechanism to two Israeli scientists back in 2004, Aaron Chechanover and Avraham Hershko. So, but the mechanism, you know, it's important also in neurons, and here it's not only in neurons, it's in the synapse. And we found that you need a protein degradation machinery to eliminate a certain receptor here in orange. But how do you get this to function in a sex-specific way? By a differential expression of another component, this thing in yellow, which is expressed in one sex, but not the other. When this is expressed in males, you see that it protects and the receptor is still active and we have synapses. When this protein is not expressed because the sex determination somehow signals to the animal, remove that protein degradation happens, and you can see that no synapses are formed. And we just published this nice work 2 weeks ago, and I'm very happy. And I want to tell you, okay, you know, you close one door and then you open many different doors. And I just want to tell you, where are we going with this receptor and why am I so excited about it? So this is a receptor, it's called DCC. We won't go in too much what it means, but it's very conserved. It's a signaling molecule. So once it's activated, it transmits information, various types of information. And what we see here is that we identified the binding site in this receptor through which the protein degradation machinery degrades the protein. So that machinery will identify the sequence, the letters you're seeing, these are amino acids, the letters you're seeing in red, and then it will know to bind this receptor and degrade it. If you look at the animals, you can see that this is very conserved throughout evolution. So it doesn't matter on which organism we look at, worms, flies, mouse, or humans, all of them have the same binding site to the same receptor. And that's really important, the key important when, in fact, when you're working or you're an experimentalist biologist, because that means we can really study one animal and then see what's the relevance to humans. And this is my last slide, one before the last. And I just want to tell you, why is it important to us specifically? So people have surveyed many humans. You can see here people with mutations. Unfortunately, you see over 200,000 humans, and they found all these people have these major psychiatric disorders, OCD, schizophrenia, ADHD, many diseases. The #1 region which was hit in all of these is this receptor that we're working on, DCC. Another study has also confirmed the same for Parkinson's disease. So it's clearly a really important receptor. We don't really understand why. And as I told you in the beginning, sex differences in Parkinson's also exist. So we're trying to link all these components together, and we know that there's differences in its expression pattern in neurons that degenerate, which is what happens in Parkinson's. And this is something which we're working on now. So I hope in a few years, I can share the results of this study with you, because they're definitely going to be beneficial to humans. And I think I'll skip that because I'm probably over time. Yes. So I'll just end with thanking the lab. We can discuss the future progress. If you have any questions, this is my current lab. These are the funding sources. Thank you very, very much for listening. And of course, I'll be happy to take any question. Just stop sharing my screen.
Meital, thank you very much for this quick and thorough presentation on a topic that for most of us here is very difficult to really understand. We love the process of closing the gap between what we do not know and what scientists like you can teach us. So we have some time now for questions and answer. And I just wanted to open up with one myself, and then we have if people would like to just write on the chat their questions, okay, please. But Meital, so do you think that for brain degeneration situations and diseases, the solution or the drugs or whatever will be different for female and males, including Parkinson, for example? You could have different treatments depending on the male of the patient.
So it's a great question. I don't know whether it will be different, but let me give you an example. We're now working with a Parkinson model. And we actually didn't think we're going to have a difference between the sexes in a drug we're giving. So there are many ways to model Parkinson. One of them is administering a certain type of chemical which degenerates the neurons, and then you have a Parkinson-like symptoms. So we didn't think we're going to have any differences between the sexes, but just looking at the way or the rate the neurons degenerated, we already saw striking differences, and that was really mind-blowing because we weren't expecting that. So it's not necessarily that we're going to have different treatments, but for example, you could maybe administer only half of a dosage to one of the sexes because the phenotype is not so severe, right? But we don't really understand yet these diseases to their core. So how can we treat the sexes differently? But this is definitely something that I think researchers should take into account from day one, because it does matter whether someone reports studies done on males or females, obviously, specifically in Parkinson, where it's not just the severity, it's also the symptoms. They're completely different sometimes between the sexes.
Thank you. We have a question here. What do you think of non-binary theories of human gender studies?
So I always get the questions of, human gender, sexual orientation. I'm not a psychologist. I can give you, right? I work with molecules and genes, but I can, give you my 2 cents. As far as we're concerned, science, in terms of the our biological sex, it's very clear. We can't be both. We're either male or female, right? Because we either have an XY or an XX. But as I've shown you, just that generates so much mosaicism in the brain that it's not that we all, we have the bell curve. It's not that we are all in the middle, but we all range, we're not all in the corners, either male or female, but we're probably somewhere in between. So I really like the theory of the mosaic brain. So I mean, yes, non-binary sounds very feasible to me when you talk about sexual orientation, but sexual orientation is so far down the levels from the chromosomes till you get there that I think many things could go different ways along the way.
Thank you. Another question is related to the animals that you are using. Can you describe more?
Yes, of course. So the major animal that we really love to work with, because it allows us to study these synapses in great resolution, because they don't have billions of neurons, they only have 300, are worms. And this is interesting because you wouldn't expect to see such striking differences between the sexes in the worms, but they do. Their nervous system is so easy to follow. that we started with studies in the worm, but we're not just doing that. As I've shown you, because the proteins are so conserved, we made the big discoveries, you know, big in quota, in worms, and now we're looking into human cell cultures and also a little bit of mouse. I prefer, you know, if we don't have to harm animals, we don't, but sometimes that's inevitable, especially when you talk about more complex diseases. But that's what I love about neuroscience. You can find it in one simple organism and then you translate it to other organisms.
Very good. The next question is about what triggers the degradation. Are there hormones that are different from males and females? What's the triggering point there?
Excellent question. People have been listening. That's a level 2 type of question. So great question. We do know how this differential pruning is brought about in the simpler organisms, the worms. So we know that they're the sexual identity of the cell. So again, I'm going back to the chromosomes. The fact that the chromosomes are different means that we have different cascades of proteins that will be expressed. And that's what happens. And then at a certain point, one receptor is blocked. It doesn't mean that in humans, it's not going to be hormonal. As I said, hormonal is non-autonomous. We don't know yet whether hormones are involved or not. Currently, we see that it's the genetic identity of the cell, meaning the chromosome complement, but I cannot rule out that we will also see hormonal effects.
Okay, very good. So people are incentivized to come with more questions. In the meantime, I'm curious about something related to your background and the description of what you mentioned about yourself. Two things. What did raise your personal interest as a scientist to go to the field of neurosciences? And the second part of the question: why did you pick Weizmann? What were the choices you had, and why did you decide to dedicate your career or to come into the team of Weizmann?
It's both good questions. Why? I don't, you know, as scientists, I think, first of all, I will say that if you choose to be a scientist, you're probably curious and interested in many, many different things. And I think we like solving puzzles and finding or connecting pieces without really understanding them, but then getting excited about seeing the full picture. Very early on during my career, it was very clear to me that I'm an experimental biologist, meaning I like to mix things and I like to see them. And I think when you think about visualizing the brain, that's always fascinating. Plus, I always felt that we almost don't know anything, right? It's a completely blank, and there will be many interesting discoveries to be made. So I think pure curiosity about something I completely don't understand was probably what pushed me towards it. But you know, sometimes you don't know why you take the path you take in life, only in retrospect. And for the second question, so it was a difficult decision and a very easy one, because I came from the Technion, and you know, my family is in Haifa, and I spent many, many years there, and I have a lot of respect for that technological institute. But when I got an offer for Weizmann, it was very clear what I was gonna do. I don't think there are a lot of places like Weizmann, honestly. You're immersed in science. The fact that I live here and I work here, it's like I'm immersed in science in every aspect of my life, and everything is close and easy, and you know, stimulating my brain 24/7. I'll give an example. So it was very easy to decide to move my family, and it was a no-brainer. I'll give an example of, you know, the uniqueness of Weizmann. I live inside the campus, as I said, and my neighbor is a chemist, and we have kids roughly the same age. His name is Nir London, and he's also a computer scientist, and we always go to play together with the kids in the playground. And we started talking about science one day, and I told you about the sequence that I've shown of the protein And we were talking, and I showed him this, and I talked to him, and then I showed him on my phone, and he's like, Meital, are you joking? Are you serious? This is exactly the site that one of the small peptides that I planned recognizes. And then, you know, next day he gives me a tube, and we started a really successful collaboration at the playground, which really crosses boundaries because he's a chemist, he doesn't know anything about neurons, and he works in cancer. So, and you know, we draw a lot of inspiration in neurobiology from cancer because there are many things that we know about cancer, but we don't know about the brain, but we can use them. And that's a perfect example of, I think, how Weizmann is a really special place.
Thank you. This is very interesting because this is not the first time that that happens. Scientists
I'm sure,
that
yeah.
are living on campus and they meet casually and they find out complementary possibilities. It's amazing. It's very interesting. Another question is, does individuals with multiple chromosome disorder like XXY or XYY or XO have the same bias on psychological disorders?
Wow, I don't know, you know, that's a really great question that I haven't thought about. So the thing is that the Y doesn't seem to play a really crucial role in the brain. And it seems to be the fact that females have 2 X chromosomes and one of them needs to be silenced because we need to have the same number of chromosomes, but some genes escape this silencing. That's the difference between the sexes mostly. So I don't know about the XYYs. I'm sure that these humans have effects on, differences in the psychological disorders. I don't think people know enough. It's not like we can take, you know, brains and image them and then say, okay, this cell looks different than this one just because the sex is different. We're not there yet in humans. We're really, really far away. That's what we're trying to do now. But it's a fascinating question, and I would be happy to read about it because I don't know enough.
Yeah. Meital, by the way, we know that now Weizmann is one of the biggest and the first priorities of the new frontiers and the new area of investments is in all the neuroscience, this big project that as a group of friends here in Brazil, we are sponsoring an important part. But why did brain research lag so much behind, you know? So of all the things that we try to understand from the body, from our genetic composition, diseases, brain looks like an empty space in terms of knowledge. Why is that? And how long do you think, if you can guess, you know, are we from the age of time when we can really be more safe in regarding Parkinson, Alzheimer, and those terrible diseases?
It's a complex question, and I think I will. Okay, so various reasons, right? I will say what connecting to what Roy has said, that we want to be where cancer is now in 20 years. So first of all, let's think about these diseases. Many of them that affect the brain have stigmas. They affect older people, which, you know, we get less money for research on that. Now it's not the case, but let's think back 20 years. Schizophrenia, right? It's a disease that people don't want to talk about so much. So most of these diseases were kind of a taboo. It's much more complicated to study the brain. And I think mostly, neuroscience was a really descriptive field until we got all these revolutionary tools of you know, the GFP and all the technology that we have, which is based on computers or in computer science, that we didn't have 20 years ago or 30 years ago. So it's nice to describe something that you're seeing, but without the mechanism, that's never going to help you, or it's not going to advance you far enough. So I think saying that maybe 20 years from now, because people are now aware that these diseases, right, our brain matters a lot, and great, we can, you know, we can cure cancer, but if your brain doesn't function properly, we want to have good quality of lives, right? We don't just want to live longer, we want to have good lives and live longer. So I think people are now realizing these, the tools are much, much better. But it's the terms of funding, right? There's only so much money to go around, and the center for many years was cancer. And I think now this is shifting a little bit. It doesn't mean that we've cured cancer, but I think this is starting to shift. And I think neuroscience is getting a lot of attention. Plus, we're getting amazing researchers that weren't classic neuroscientists, right? Computer scientists or theoretical or mathematicians. They weren't the classic neuroscientists 20 years ago, but now it's very, very clear that we need them to understand, because the brain is complex, right? And we need these people for the complex parts of the research.
Another question comes, thinking about epigenetics, do you believe that gender identity can modify these patterns of degradation?
I don't know if specifically the degradation, but definitely we know, and people have been studying it mostly, I think in the US, that there are epigenetic differences between the sexes. That's known, and people are studying it, but that's earlier on, right? So we're talking about, think about it, different phases of development. So in embryogenesis, and then during sexual maturation, development, and then maintenance as adults, that's a really important part, and then aging. So definitely epigenetics plays a part. I don't know if that's gonna affect specifically the degradation, but it plays a role. Degradation doesn't explain everything, right? This is just one mechanism that we found to see how we get differential connectivity, but there are many, many different ways to do that. So the short answer is yes.
Meital, in the next 5 minutes, I have my last curiosity question, and then I will close and then conclude the event. But my top curiosity is, do you have any recommendation? Can we do anything to be more preventive in defending our brains from degrading? Or this is just something that the science world does not have any hint about.
It's a good question. Also, I can, we cannot solve all diseases, but we do know that genetics is a small fraction of these diseases. It's not that all of them exist if you have the mutation. No, there are many, many different factors. And I think we know that, at least for Alzheimer's, if you work your brain all the time, you use it, you don't just watch TV, but you stay active for a long, long time, and until you're much older, then your brain is less likely to degenerate. I think people that are active until, or work until their later years, suffer less from Alzheimer's. So I think it's just, of course, it's all correlative, right? But I think if you stay active and you engage and you stay stimulated, brain-wise, that's if we exercise our bodies, we of course need to exercise our brains as well. I think I read somewhere that scientists are less likely to get Alzheimer's as well, not because they're special, but because I think they think a lot. So I think that would be one recommendation.
Very good, Meital. I want to thank you very much. In closing here, I would like, first of all, to congratulate Weizmann. We are excited with this new projects in neuroscience. We are excited with the new news about the connection with the United Arab Emirates. I would like also to congratulate you, Roy and Weizmann, for hiring a scientist of the quality of Meital. And of course, congratulating Meital to having accepted and joined the family at Weizmann. This always gets us more excited. The more we have this is the third of a sequence of lectures that we got, an excellent level. The more we have it, the more we know how much we do not know, which is fascinating. So it's a privilege to have the opportunity to be connected with an institution like Weizmann, that every contact we have with people like Meital, like Roy, and the other scientists that were with us, we only have very intelligent, interesting, and learning experiences and conversations. So it's fantastic to be connected with Weizmann, and I hope everybody here had a chance to enjoy it. And now I handle the mic to Danny to conclude the event. Please, Danny.
I just want to say thank you, Mario, and Shana Tova to everyone.
Thank you.
Thank you very much, Meital. Thank you very much, Mario and Roy. We are very happy that we are closing the CERS event. We spoke about stimulating a series of science. And I have to tell you the truth. At the very beginning, we did a mistake. We wrote simulating and not stimulating, but this is not simulating. This is a real issue. This is science from the Weizmann Institute, and we want to thank to all the scientists. Now, I hope you enjoyed the series, and we are looking forward to see you again in our international board in November, November 15. We will send you information, and it will be great if you can participate. This time, it will be through the computer, but we need to keep the communication, and it's very important for us that you will participate. In the meantime, I want to say Shana Tova and Metuka, and I will open the microphones. We finish right now, but I will open anyway the microphones if someone wants to say something. You can unmute yourself.
Say congratulations to Meital. Oren, it was a great conference with not too many scientific data, but with a very good idea and great idea. So thank you, Danny. And of course, Mario Fleck, that it's now and ever a very important leader in Brazil and for the Brazilian community. I wish Shana Tova to all. And I know that the good things that will happen next year is that we will continue to have these events, and we will try to participate on them. Thank you, Mario, Danny, and everybody that is involved in these conferences.
Thank you, Regina. Regina is the #1 brain of this group. So talking about brains, Regina, we enjoy very much the connection with you, and we wish you also a happy new year, Shana Tova, and all the successes in your research here as well.
Regina is Regina Marcus, our Professor Regina Marcus from Sao Paulo, our Vice President in Brazil.
I also want to congratulate the Weizmann, our researcher, and Mario Fleck for a wonderful talk, and Shana Tova Lekulam. Shalom al Israel. Shalom al Israel.
Shana Tova, guys, thank you very much for joining in. Hope to see you soon. Cheers.
Muito bom, Dani. É isso.
Ok, muito obrigado. Shana Tova, toda rabá Meital.
Tchau. Bye bye.
Shana Tova.
Shana Tova. Ciao, ciao.
Bye, Shana Tova.